Hi, everyone. We'd like to start. Thank you, everyone, for coming. I'm very happy to introduce Shira Knafo for today's seminar. Shira is both MD and a PhD. She got her degree from Ben-Gurion University. And after an internship at Soroka Medical School, she did a postdoctoral research at the Cajal Institute in Madrid. And then she started her own lab in Basque Country. And since 2018, she's been the head of the molecular cognition laboratory at Ben-Gurion University. She's particularly interested in the molecular and cellular basis of cognitive function, and I think that's what we're going to hear today. So thank you very much for coming all the way here.
Okay, thank you very much for inviting me and for giving me the chance of spending Christmas in Jerusalem. So today, let's see if it works. No? על מה צריך לכוון כדי שזה... איפה המחשב פה למטה? קודם עבד. אולי פשוט נוציא את ה... הנה, בסדר, אני אעשה בעלה.
הקליקר לא עובד?
עכשיו עובד, אז מה קרה?
הגעתי להסתדר.
Okay. So I've been studying learning and memory all my scientific life, and the reason that I'm interested in memory is because there are many conditions that involve cognitive impairment or memory loss, and they include the obvious ones like Alzheimer's disease, Parkinson's disease, but even conditions like schizophrenia and depression, they also include some component of cognitive impairment. According to Alzheimer's Association, 1 in 9 people will develop Alzheimer's disease, and we can see that with the time we have an increase in the incidence of Alzheimer's disease. And we also know that unfortunately there is no treatment for Alzheimer's disease. We're probably thinking, about the FDA approved drug, and it's a topic of another lecture. But basically today there is no treatment. So I'm trying to develop new strategies to discover new cognitive enhancers. So I'm going to tell you a little bit what I did so far and how it gets to maybe a way to advance faster in the discovery. Just a summary of what we have so far. At least in Alzheimer's disease, we know that there is neurodegeneration or neuronal death. We don't have a solution for that yet. We know that there are accumulation of amyloid beta and in plaques and in a soluble way. We know how to get rid of the amyloid beta. So this is the new drug that was approved by the FDA, a vaccine to remove the amyloid beta, but it doesn't seem to improve cognitive impairment. There is also another protein that accumulates, is the phosphorylated tau, tangles, that causes neuronal death. Still, there are some clinical trials working on it, still no solution. Then there are some drugs that increase the amount of acetylcholine or block NMDA receptors that they work for a few months, patients with Alzheimer's disease, but they don't look for, they don't always work, and usually it's only for a short time. So what I'm trying to do is to start from an idea. An idea usually is a very basic science. We study molecular mechanisms of learning or memory. We try to work from the molecular to the system level. We discover some discoveries, very basic discoveries, and we try to convert it to drug development, which is a very long process, can take years to do this cycle. But the final goal is eventually to find therapies and to help people. It's not so easy. My inspiration and motivation was from a paper published by Danny Selko in Science. And he was claiming something that at that time, we're talking about already 20 years ago, it was completely innovative. He was saying that Alzheimer's disease is a synaptic failure. And when I was studying in the university, we learned that Alzheimer's disease is a neurodegeneration and it's plaques. And this is what we see in the microscope, and this is why we have Alzheimer's disease. But he was claiming in this paper, before he really had evidence, that the synapses are the ones that are failing in Alzheimer's disease, and they fail at the beginning of the disease, before neurodegeneration and plaque accumulation. So basically, you all know what is synaptic failure, so I don't have to explain, but we're talking about the problem of transmitting the information from one neuron to the other. The synapses are not efficient, okay? And I'm going to concentrate on this, especially on AMPA receptors that represent one of the receptors in the excitatory synapses. And especially what is interesting for me is the synaptic plasticity, the long-term depression, where you see the removal of AMPA receptors from the postsynaptic compartment. They go through endocytosis, and you have the, let's say, the opposite phenomenon that is called long-term potentiation. You all know what I'm talking about. In long-term potentiation, the final mechanism is the delivery of AMPA receptors to the synapses, what makes them stronger and makes the synaptic more efficient. And we know that this has been studying for a long time, that the AMPA receptors, they determine the synaptic strength. So, sorry, if you have more AMPA receptors in the synapses is stronger, okay? And not only that, AMPA receptors are the one that participate not only in long-term potentiation, but also in relearning, because if you train mice in different kinds of learning, in this case, acute reconditioning, you can see increase in AMPA receptors in the synapses. Of course, this is not a general increase in all the synapses. It has to be in relevant synapses. Synapses are relevant for this learning or participate in this learning and memory. And you can do that with endogenous receptors, but you can do that also with recombinant receptors. These are AMPA receptors that are tagged with, in this case, EGFP. You can see that they behave like endogenous receptors, and they are delivered and incorporated into the excitatory synapses upon learning. So this is before, and this is after learning. In LTP, you see the same thing. You see this is the baseline, the normal recording of LTP in the lab. You can see that after induction, that could be done in different ways. You see an increase in the synaptic efficacy or strength, and you see that there are more AMPA receptors in the synapses. So I'm going to talk about two drugs that we know that do at least two things. They enhance learning and memory on one hand, and on the other hand, they enhance the amount of AMPA receptors in the synapses. Okay, and for example, one of the drugs that we already worked on a long time ago is FGL. FGL is a non-cognitive enhancer. Already it's been known for around 15 years. It's one small piece of the neurocellular division molecule. It's endogenous protein, very important protein in the brain. There's only a small part of it. It's around 15 amino acids that we know that activates fibroblast growth factor receptor 1, and this activation causes phosphorylation of this receptor, and then downstream activity of different signaling pathways. All of them increase the amount of AMPA receptors, but we found out the specific pathway that is activated by FGL. And this is just an example of water maze with, in this case, we did it with the rats, and we injected them with FGL intraperitoneal injections, so 3 times before the training, and we saw a very good learning and memory that lasted even 2 weeks after the injection, and this is because of activation of signaling pathway that get auto-activated and they continue to be activated for at least 2 weeks. It's important to say that FGL passes the blood-brain barrier, and it's a peptide that is quite unique that passes the blood-brain barrier. And this is actually our prototype of good cognitive enhancer. The nice thing about FGL that it can improve memory and learning in animals that don't have problems of learning and memory. We're talking about completely healthy rats that have perfect learning and memory, but FGL proves that we can improve even and improve learning and memory. So we can, our brain is not active to its maximum level, or the synapses are not the best that they can be, and we can improve better the learning and memory. By the way, when we published this paper, people actually bought in the black market the FGL and started to inject it to themselves, which so that's the cost of publishing in open access journal. So basically how we found out about what is, sorry?
What did they report?
They said that suddenly they feel that the brain is very clear and they can think clearly, and it's completely different from rats, so it's not, it was not well quantified.
What's the stock market price?
Okay, the FGL I can talk to you about it later, what happened with the FGL. But basically FGL is a very interesting drug, because what it's doing is, this is a mechanism that we discovered, is that it's enhancing the delivery of AMPA receptors to the synapses. And this is why the connection between AMPA receptors and the cognitive ability. We did that with rectification assay. We determined the ratio between the AMPA current in the -60 and +40 voltage of the memory potential. And the way that we do it, we infect neurons with a special virus that we use called Synvis virus. This virus likes to infect only neurons, which is an advantage. And we can see here AMPA receptors tagged with EGFP. And this is only to find out where are the infected neurons. But in fact, the AMPA receptors that are tagged are not in the synapses. To go to the synapses, they need a strong stimulation like LTP or learning. And we found that with patch clamp, we found that FGL increases the amount of AMPA receptors. This is we did with electrophysiological tagging. This is just as a background. Okay, so basically, a mouse or rats after FGL injection, they have more facility for AMPA receptor delivery, and they can also learn better and have better memory. So basically, we found the pathway that is doing that through activation of PKC pathway, then CAM kinase 2, and phosphorylation of AMPA receptors, and then enhanced delivery of AMPA receptors. This is on one hand. This is FGL, and now I'm going to tell you this FGL with healthy animals, and I'm going to tell you another drug that also enhances the amount of AMPA receptors, but in this case only in models of Alzheimer's disease. Basically, we are talking about a drug that acts on a specific pathway called the PIP3 pathway, and we know that it has 2 enzymes that control the AMPA receptors. It's the PI3K enzyme and the P10 enzyme that do the opposite things. If we overexpress PI3K, we have an increased AMPA activity in the synapses, and if we overexpress P10, which is the opposite enzyme, we have less AMPA in the synapses. So basically, this guy contributes to LTP, and this guy contributes to LTD. Okay, and not only that it contributes, we can see that if we block P10 is the one that contributes to LTD, we don't have LTD, and this is we can do that either with a drug or with the dominant negative form of P10, and we can see that P10 is very important for processes of LTD. So this for us was a sign that we have to check this enzyme further, and the reason is that I was trying to combine what I knew about AMPA receptors, about cognitive enhancement, and try to see what we can do in the case of Alzheimer's disease. So as I said before, there are several drugs that are aiming to target amyloid beta. Probably you know about the amyloid cascade hypothesis, saying that amyloid beta is the cause of Alzheimer's disease with a cascade that starts with the overload of amyloid beta and ends by neurodegeneration and synaptic failure. But many people challenge this hypothesis now because they think it's too simplistic. And also we know that even if we remove amyloid beta from the brain, we still don't see a real improvement. So what can we do? We were thinking of doing something else, a completely different idea, in which we decided to target not amyloid beta. We want to leave it alone. We want to leave it in the brain. Okay, we quite ignore it. Okay, what we want to do is to target the synaptic depression. Okay, we know that Alzheimer's models, there is a tendency to develop synaptic depression and really have big issues in generating synaptic potentiation. So we said, okay, let's tackle the issue of synaptic depression. We try to prevent synaptic depression and see what happens. One of the most important experiments that we did was to do time-lapse imaging of P10 tagged with EGFP, put amyloid beta, and see what happens to the P10. And we saw that quite immediately, we saw that P10 enters to the spines and stays there. Okay, if we express another kind of P10 that lacks the C-terminus, which is the PDZ binding domain, we see less entry. We thought that here we have something because we can have a mechanism of synaptic depression in Alzheimer's disease. And because we know that P10 is so important for depression. Okay, so basically we published in that paper that P10 is more in synapses in primary neurons. Years later, we also confirmed that in brains of Alzheimer's patients in different stages, and we see that the more the advanced stage of the patient, we see more P10 in their synapses, which we checked with around almost 200,000 synapses, and it's quite strong evidence. And what we see basically what happens in Alzheimer's disease or in models of Alzheimer's disease is that P10, when we have amyloid beta in the area, P10 enters the spines, interacts with the protein called PSC95, and this is what causes the synaptic depression. Just to make a very long story short, okay? If we block P10 in cultures, in organotypic cultures, sorry, you went too far. We can see that we can recover the synaptic function, and this is without getting rid of amyloid beta. We have amyloid beta and we see even overshooting of the synaptic function. So we did that with a neuron-derived amyloid beta, which is very, very toxic, and we did that with a synthetic amyloid beta. It slices, a different technique, but the result is the same. If you can block P10, you can recover the synaptic function. And the next step, of course, was to do free conditioning. One of the tests that we did, inject before for 3 weeks the P10 inhibitor, and later the P10 peptide, and to see that injected P10 inhibitor has normal memory, drug, they don't show any memory. Okay, so the next step was to develop a drug that can prevent this interaction between P10 and PSC95, And we developed a small peptide that was exactly the C-terminus of P10, the part that is the PDZ motif, the one that's supposed to interact with PC95 under pathological conditions. And indeed, we can see that we can rescue the synaptic function and the cognitive function. So basically, this is what the drug is doing, interferes with this interaction. This Memory Plus is the company that we have now to get this drug to clinical trials, but it's another story. And all this, I was telling you to get to the other issue that is how to use this knowledge that we have until now between the strong link between AMPA receptors and the cognition in order to develop new drugs. Okay, so I told you about 2 drugs. Of course, it's not enough. We need to have many drugs, so at least one of them will finally get to the clinical, to the clinical trials and then to use. And for this, we need to develop an assay that can actually screen these drugs, and get to the appropriate ones based on their ability to affect the AMPA receptors. So this is basically, it's based on this. We want to start with the culture, even in a 96-well plate. We want to use a plate reader, which is much simpler than doing electrophysiology, and then do the processes of drug development that we already have experience with that. And finally, this is the final goal, to get to the patient. Okay, so we had to develop a new, completely new method, okay? And for this, we use some of the things that were already existed, put them together, but some of them we developed ourselves. For example, I already told you about the EGFP GluA1. So instead of EGFP, we use sept GluA1. This is a super ecliptic fluorine that is EGFP that the fluorescence that it emits depends on the pH. So if it's on the surface of the cell, you will see it. If it's inside the cell, you don't see it. It's perfectly good for identifying synaptic AMPA receptors. So this is before stimulation, the same neurons, and after stimulation of the synapses, you can see that big difference in the fluorescence. Okay, this is because the AMPA receptors is now in the surface of the cells. Okay, so we can, the idea is that if we have this tool, we can detect drugs that enhance the synaptic transmission, okay, without electrophysiology and without imaging, okay? So it was quite a lot of work because we had to adapt normal cultures that we do in the lab to a different format of cultures. This is 96 well plate cultures. So we had to adapt a few things. We had to make sure that the neurons look the same, that, for example, they have the same spine density. So we had to study that to find the optimal conditions. We also wanted to know how much glia do we have in the cultures, and to control that. We found out, for example, that half of the cells in the cultures are glia, okay? And we can control it, but we tried experiments without glia, with glia, and we found out that this is a good combination of glia and neurons. We had to make sure that our virus infects at least 90% of the neurons, if not 100% of the neurons. And luckily, our SynBase virus, when it's done, when it's prepared appropriately, it's a very effective virus and can infect 100% of the cells of the neurons. Okay, not the glia, and we did that with the fax. We also had to check that we can indeed, with the plate reader, we can really detect changes, subtle changes in the fluorescence in the culture. So it's in this, every square here represents one culture, okay, in a 96 well plate. And depending on how much virus we put, we can see more fluorescence. Okay, so the plate reader, it's not a normal plate reader, I have to say. It's a plate reader that has an incubator and humidity control, and so it controls the temperature and the CO2. So it's more sophisticated than the usual, but it's a very, very sensitive plate reader. And another thing that we had to make sure is that we can see drugs using the green AMPA receptors with the set, but we also wanted to have a red version of the same receptors. Why is that? Because what we saw is that many drugs has autofluorescence that is green, and we wanted to be able to screen also in red. And the other thing is that the medium that we use, the medium also has some green fluorescence. Okay, so if we use the red fluorescence, we have less of this problem as long as we get rid of the phenol red in the medium. So we made a completely new construct that is Fuji instead of set, Fuji Glue A1, so it's in red. Okay, but the idea is the same, that they respond to synaptic activity by increasing the fluorescence, increasing the amount of AMPA receptors in the synapses. What is also important to say that we are talking about the conditions of overexpression of AMPA receptors. We have not only that we have the recombinant AMPA receptors, we have also the endogenous AMPA receptor that is higher. So we have around 3 times more AMPA receptors than the usual. Okay, and this is the way it looks. So we can change the pH, and we can see a decrease in the fluorescence when we decrease the pH, which is what we expected, but it was not easy to get to this point. We had to optimize the medium where we scan with, of course, we don't have any background with the water or PBS, but we have background with the medium, which is lower when it's red fluorescent proteins. And this is a robot that we have in the lab. So we can see, we wanted to see that we can see expression of recombinant protein with the plate reader. Indeed, this is a expression of DT tomato, and you can see an increase as the time passes. Okay, and then something very important that we had to do. If we want to see that the drug enhances the delivery of AMPA receptors or induces more AMPA receptors in the synapses, we have to make sure that we know how to induce that. So we already said that LTP induces delivery of AMPA receptor to the synapses, but we do that usually we induce LTP with the electrical stimulation, and here we had to induce it with chemical stimulation. So everybody knows how to do LTP. It's chemical LTP. It's published, but it was not so easy to make our culture, 96-well culture, to induce LTP, and we had to do a lot of biochemistry to make sure that we indeed induce LTP. The way that we do it is that we use markers. So we know that during the chemical LTP, we have more phosphorylation of AMPA receptor in specific amino acids. So we know how to check it. We also know how to detect LTD with biochemical tools. So we had to optimize the LTP protocol. And then, of course, the question was, can we see LTP with a plate reader without electrophysiology? So this is the way it looks, just normal microscope before and after LTP. You can see that you can see more fluorescence. Okay, now the question, this is with the microscope. The question is if you can see it without a microscope, only plate reader, just the fluorescence. And the way that we did it is just induce LTP with different concentration of glycine, which is a normal LTP protocol. And we saw the induction of LTP. So if you have experience with LTP, you know that when you do electrophysiology, it looks exactly the same. And we can see that it depends on the amount of glycine that you put. And if you want to compare normal LTP experiments that I did and many other people did, you know that it takes time, either in a patch clamp or field recording. It takes time for one drug. And it takes 2, 3, 4 weeks to finish one drug. With this system that we call Fortis, it can take 1 hour. 1 hour for 30 drugs. Depends how many repetitions you want to do. So it looks the same. We tested later with different concentration of glycine here in hippocampal neurons. We can see the difference according, we can see dose response, actually LTP, according to amount of glycine. We did that with cortical neurons. We saw exactly the same thing. We then check how much, let's call it LTP, how much LTP we have. This is frequency distribution. We see that it's most of them are around 30–40% of increase, which is very similar to what we see with electrophysiology. and it's very similar between the hippocampus and the cortex. So people that have problems in taking out the hippocampus from embryons, so the cortex is also good for this. And I was also thinking that it could be nice to also confirm LTP in animals that we know that have LTP impairment. So in this case, we took an article that we published in Cerebral Cortex 2 years ago. Mice that overexpress PTEN, they show impairment in LTP, and not surprisingly. And when we use the same mice, we take embryons, we make from them the cultures, and then we induce LTP, we also see the same impairments in the fortis without doing electrophysiology. So this shows that this system, the fortis, can be used not only for drug screening, but also for experiment, normal experiment phenotyping. So it is a very fast way of phenotyping mice. We can also detect long-term depression because we know how to induce LTD with the NMDA, application of NMDA on the cultures, and we can detect this with, again, phosphorylation of AMPA receptors. And we can see here a decrease in the fluorescence when we reduce LTD. So now the moment of the truth arrived because we wanted to know that this was the purpose of the development of fortis, that we can actually detect drugs that we know that are cognitive enhancers, and we know that they act on AMPA receptors. We don't expect it to detect all the cognitive enhancers because there are many ways to enhance cognition. Some of them go through AMPA receptors and some of them not. Okay, some of them go through NMDA receptors or other receptors, but fortis is meant to discover the ones that go through AMPA receptors. Of course, you can adapt it to other receptors. If you want to make a clone and other receptors with CEP or Fuji, it's possible too. Okay, so this is the result of FGL that we did with past clamp. We saw an increase, for example, in LTP. We saw an increase also in the basal synaptic transmission, the AMPA current. And with fortis, not only that we can do the normal dose of FGL and we see an increase in this dose, but we can also do a dose response. Why? Because it's easy. It's not patch clamp. We can do a lot of the concentrations. We don't have to think twice about it. We just put different concentrations and we do the dose response of the fluorescence. And you can see the fluorescence in response to FGL. Of course, the next thing was to check the PTEN peptide that we published a few years ago, that we saw that, first of all, in the paper that we published, we and others saw that if you put amyloid beta on cultures, on slices, or even in animals, we see synaptic depression immediately, okay? We wanted to see, this is with patch clamp, and neurons that overexpress or overproduce amyloid beta, you can see a decrease in the AMPA currents, okay? And the parallel of this would be a decrease in the fluorescence in the fortis. So what we did is very simple. We took neurons, we made them to express or overproduce amyloid beta, okay? Then we took the medium of these neurons and put it on other neurons, the neurons that are in the 96 were plates, okay, that were already infected or expressing CEP-GLO1, Fuji-GLO1, okay? And you take this medium and you get a lot of concentrated amyloid beta, very toxic amyloid beta. And what we can see is that there is a decrease in the fluorescence after we put, this is 24 hours after we put amyloid beta, and secreted from other neurons. Okay, so we see that both in the, we see that in with the green and with the red, I mean with the CEP and the Fuji, we see the same effect. So first of all, it was important to see that we can actually detect endocytosis of AMPA receptors after amyloid beta, and this is the basis of later to develop drugs that can prevent this from happening. And then the issue was to use the peptide that we developed, okay, and we saw that it can prevent synaptic depression, and to put it in the fortis, okay, and we can see that the more peptide we put, we see a better fluorescence or more fluorescence. Not only that, we saw that the peptide can rescue LTP. I'm talking about chemical LTP with the fortis, so we induce LTP with amyloid beta, you won't see almost any LTP, but with the peptide that is a cognitive enhancer, you can see more LTP depending on the concentration of the drug. So basically, we were able to detect 2 cognitive enhancers with this system without using electrophysiology, but let's say repeating the experiment that we already did with electrophysiology. So we knew what to expect and we got the same result. But this is apart from the fact that we had to actually develop this method, which is what was quite challenging. Once we have it under control, the experiments are not very difficult. And so we have a system that is quite fast because we can do, I won't say that it's high throughput, but compared to patch clamp and field recording, it's much faster. It's cost effective because you don't have to buy expensive electrophysiology equipment. We can screen, well, I think it's very optimistic, but it's possible if you have enough people working on it, 15,000 drugs in 5 years. And we expect that if we do a systematic screening of drugs, of candidate drugs, we can maybe find a few drugs. And so this system is, we designed it specifically to develop, to detect drugs that are possible cognitive enhancers. And we can, I think that it can lead to develop of a series of drug and maybe advance the field of cognitive enhancement that I think is very behind in many aspects. And this is what I did. The agencies that actually had to perform these experiments, and happy to answer questions. I hope I didn't go too fast, but you can ask questions. Yes.
Yeah. So this is really fascinating stuff. I was wondering what early phase of long-term plasticity and late phase. Right. So these drugs, definitely the P10s, that seems to be targeting the late phase. At least I saw in an LTD plot that you had. So you have the initial sort of depression, and then but when you right, so when you don't have the P10, it like goes back to baseline. Right? So can you talk a little bit more about which part of the plasticity you're targeting?
We didn't check plasticity for more than 1 hour, 1 hour and a half. So I cannot tell you about the long term. What I can tell you is that when we treated the mice for 3 weeks with the mini-osmotic pump that releases the drug, it's either a PTEN inhibitor or the PTEN peptide. When you prevent LTD continuously, it has an effect. I don't know for how long it acts. So I mean, I don't know if I inject it now and I leave the animal for how long the effect will last. I don't think it will be very long. Okay, that's why we injected it with the pump. Unlike the FGL, that's the other drug that we know exactly the mechanism that it acts, that it activates the kinase 2, that autophosphorylates itself, and then it can last for at least 2 weeks. And this is a completely different mechanism. But the final result is that it acts on AMPA receptors. The idea is to keep them in the synapses as much as possible.
Your model of the disease, though, is can you like summarize that? So Alzheimer's is that you have too much LTD, that LTP is not being stabilized. Like, how would you frame that?
Alzheimer's disease, it's a very complicated disorder. Okay, so many things happen there. You have changes in inflammation and in glia and anything that you can imagine that you have problems with that. We are studying the part of the synaptic function, okay? And we know, and we saw, and other people saw, and not the first, that the synaptic function is particularly impaired. If it's probably it's one of the later things that happen, I mean, in the cascade, okay? And we are trying to kind of ignore the rest of the things that happen and go directly to them. Directly to the synapses.
Within the synaptic part, right? So which part of plasticity are you saying is the part that Alzheimer's?
What we saw in Alzheimer's, in models of Alzheimer's disease, that you definitely see less LTP or no LTP at all, more LTD, less basal synaptic transmission, I didn't see an effect on the presynaptic function. And we're trying to revert that. We are trying to encourage the AMPA receptor to go to the synapses and incorporate into the synapses. So we have more LTP, less LTD, okay? But this is the balance that we are trying to change, okay? In the animals, it works. But to tell you if it will work in humans, I don't know. We'll see. Yes, more questions.
What extent is the high throughput externality is general in the sense of what is it? Very specific only to, I mean, obviously that's what you quantify. Can it detect synaptic dysfunction?
It will detect everything that eventually depends on AMPA receptors as the endpoint. It can be all kinds of things that go wrong in the cell because you know that there are many signaling pathways that eventually lead to delivery of AMPA receptors or endocytosis of AMPA receptors. It's a whole thousands of molecules that are involved in that. So these molecules, if you can manipulate them, if you can change them and they change the AMPA receptor at the end, you will probably see it in 40s. If it's not expressed as AMPA receptor changes, probably you won't see it. But as I said, you can clone any of your favorite receptors and try the 40s on them. Not so complicated. You just have to attach it to the pH dependent tag. Okay?
You try no damaging agent leading to synaptic damage.
What do you call synaptic damage? That they become weaker, that they shrink, that they disappear. I guess that if they become weaker, you will see it. If they disappear, you probably will see it also. If they shrink also, because if they shrink, usually it means that they also have less AMPA receptors.
I mean, that's where does the fluorescence go? Right?
If the fluorescence is in the cytosol, probably it's you won't see it so much as if it's in the surface.
If the fluorescence was already activated, then it gets phagocytized.
You won't see it. Okay, it's a dynamic thing. You can see it, and then if you have endocytosis, you won't see it, or you have see much less. Okay, more questions? I can't understand anything.
If you basically keep a lot of adverse effects on the surface, I'm wondering if this senses that anyway transcriptionally, or
That's a very good point.
overall adverse effects.
That's a very good point because you are asking about for each individual, if I understand, for each individual cell or synapses, you have the basal conditions. So you can have more or less AMPA receptors already. Okay. Of course, this can affect the transcription of different molecules that we don't check here. Okay. But what we found out very interesting is like similarly to what you do with electrophysiology, that you see that when it's easier to induce LTP when the synapses are weaker. It's kind of called metaplasticity. You can also induce more increase in fluorescence when you start from lower level of AMPA receptors, from lower level of fluorescence. That's why we were very careful not to express the recombinant AMPA receptor too much. Because if we express it for 2 days already, you don't see any LTP. Okay, so it's a the balance is very important. You want to keep the neurons on one hand fluorescent, on the other hand, not too fluorescent that you won't see any expression of LTP. I hope I answered your question. Okay.